Thank you, everybody, for joining us today. We're just going to give people a minute to get into the room, and then we'll get started. I'm going to get feedback from yours while we're talking. Can you just mute for a minute?
My challenge is, oh yeah, you're right.
Just for the minute to keep it going. Can you find it or do you need to
I
Okay, welcome everybody. In the interest of time, we're going to get started. Hopefully a few more people will come in. So hello and welcome to Science by the Slice. We're doing this as a hybrid event. I'm Fenella Saunders. I'm the editor-in-chief of American Scientist magazine. These talks are put on by Science Communicators of North Carolina in collaboration with the RTP chapter of Sigma Xi with funding from the NC Biotech Center. So thanks for joining us today. Today we have a really great talk from Sid Thakur, who is going to talk to us about One Health approach to understanding the complex dynamics of antimicrobial resistance. So Sid, I'm going to unmute you now and hopefully you can take it away.
okay.
Hold on a second. I had to ask you to unmute. Is it unmuting you? Let me see if I can do this.
No.
Oh, come on. I don't know what it's doing. 1 second, folks. We got to get Sid unmuted here.
Got it.
Great.
Good to go. All right. Thank you all for inviting me. I really appreciate it. So just a fair warning, we are going to talk about antimicrobial resistance in foodborne pathogens while you're having your pizza. And I hope my talk is not going to be like the weather outside, but it's a complicated topic. It's a very important topic based on the diversity of the audience in terms of their interest and background. I'm going to float at different levels. All right, I'm going to big picture, then I'm going to show some more fine-tuned data in terms of more granularity, and hopefully we will have fun. And so this talk is more of my whole, I've been working in this area for more than 20 years, all the way from a grad student. And it's sort of my thought process as it has also evolved over the last 15-20 years on this very important topic. And it's a global problem, it's a complex problem, and I'm going to talk a little bit about One Health, what One Health means and why this is one of a very interesting topic for us to solve. Okay, so that's the first problem. And let me see if I can do this. Okay, so just a little bit about my own background. I work a lot with food animal productions in North Carolina. As you know, North Carolina is very big in pork and poultry, and I use what is called a systems approach. So I'm not just focusing on the animal. I'm also looking at what's in the environment, what's in the population around it, and believe it or not, this is the same slide I used 16 years ago when I applied for an assistant professor position at NC State, because it has not changed. The focus has not changed, but the sort of the span and the scope has changed. So real simple, we do a lot of on-farm sampling. We go to the farms, we collect samples, environment. We also work with the state public health lab in terms of what's happening on the human side for these infectious pathogens, targeting bacterial foodborne pathogens. We bring these samples back to the lab, we isolate different pathogens, then we figure out whether they're resistant to different antimicrobials that are used in animals and humans. Once we find resistance, we go deep inside where they are located, those genes. And then, of course, we do a lot of genomics. So all the way from farm to the genome, that's why I call it the farm to genome approach. But one thing that has happened over the last few years is I'm not just interested in what's happening with these E. coli's or salmonellas in North Carolina. I'm really interested to look at it from a global point of view, because pathogens behave in a very different way based on the pressures they feel in different geographic regions. So, when it comes to resistance, if you want to go to CVS pharmacy, you need to have a script. In many parts of the world, you don't have to do that. You can just go to any pharmacy store and get any antimicrobial you want. Okay, and so when pathogens are exposed to extensive amount of drugs, they react in a very different way. Okay, so E. coli in the US may be very different than E. coli in, say, Southeast Asia. And the basic take-home message at the end would be, it's a very simple concept of how pathogens evolve is the Darwinian principle of survival of the fittest. It really absolutely applies really well over here. So the other thing that we do in my lab is we run big surveillance programs. The first one is they're both funded by FDA. So FDA's Genome Tracker program is generating whole genome sequences. Every year we generate anywhere from 400 to 500 sequences a year. And the second one is called the National Antimicrobial Resistance Monitoring System. So the retail meat that you and I buy from supermarkets, my lab, in collaboration with some other labs, generates the resistance profile for different pathogens for the entire state of North Carolina. Okay, and so that data goes to FDA, and then FDA, CDC, and USDA come up with the combined report every year. So I can tell you what we find, for example, in the retail meat that you and I buy. And so a few years ago, we started seafood sampling also. And that's wild, wild west in terms of resistance in seafood. Okay, so I really like the One Health model. That's also the Global One Health Academy that I lead at NC State, that was established a couple of months ago by the provost office. And One Health is the idea that, all the health, whether it's plant health, animal health, environmental health, they're all it's the same. You cannot really improve one health before you improve the health of everybody else. All right, so it brings everybody, people with different disciplines together, different backgrounds together to work on big problems that need a multidisciplinary approach. Okay, and so even if you look at the funding model in the US right now, the days of single PIs don't exist a lot. Funding agencies want interdisciplinary approach, people with different disciplines working together. And that's where One Health really flows very well. So amazing example is the pandemic, right? And so, exchange, something happened in the animal side and then trickled through the environment in the human side. And the concentration was so high that, everybody's spreading it around because it's, air to air. And there's also reverse zoonosis, we are saying, because the concentration in humans so high that, minks and, ferrets can also get infected. And so right now, the focus has always been for the last couple of years on vaccines, making sure everybody gets vaccinated. But the One Health approach is going to soon trickle in, because ultimately we want to go back to the source. And because who's to say that, there's not another virus that's, working its way through the system, right? So we got to stop it at the time, right where it starts. And for that, you need veterinarians, all right? You need people, environmental scientists. We have a surveillance program also looking at, SARS-CoV-2, the virus and wastewater, right? And so we need an approach like that. I don't think there's a better topic in One Health than antimicrobial resistance, okay? Because we find it in humans, we find it in animals. It's a big environmental reservoir. I've been working in this field for 25 years. I don't remember the last time I heard a good news when it comes to AMR, and I'll make a case for it because it's a very complex problem. But it's impacted by climate change. Rising sea temperatures have been associated with the increase in prevalence of resistant pathogens flowing through the communities. Wastewater production plants, use of antimicrobials, and of course, a big reservoir in the environment. So you need a holistic, sort of big picture approach over here to work together. So I'm just going to call antimicrobial resistance as AMR moving forward. So AMR is a global problem, okay? So let's look at the US right now. So if you look at the estimates, 35,000 deaths every year because of drug-resistant pathogens. But whenever it comes to talking about numbers of people infected and dying, there's a massive amount of underreporting, okay? So how many times, we are not feeling too good, who's going to go and to my physician, I just take it easy. Well, you're an underreported case, okay? So based on the pathogen, you can multiply whatever number you see by 18 to 34 times and to get a real approximation of how many infections are caused by a particular pathogen. Significant impact on animal health, and also a big pressure on our public health infrastructure. So patients who come to hospitals with drug-resistant pathogens, they spend on an average 3 to 5 days more in the hospital. It just takes longer time to resolve the situation. And that just adds up to the cost. Now, when you look at the global level, outside the US and outside the European Union, the developed countries, the situation is really bad. Okay, so if you talk about hot zones of new emergence of these resistant pathogens, typically Southeast Asia, Africa, because of misuse of antimicrobials, or actually no jurisdiction in terms of, how and where you can buy it. So again, pathogens are stressed. When they're stressed, the Darwinian principle of survival of the fittest, and then they land up on a front door very soon. And COVID pandemic is a very good example of that. So if you look at the Lancet report that just came out, that was giving us an estimation of how many people died in 2019, it's around 4.8 to 5 million people. Okay, globally, yes.
Is that excess deaths or total deaths?
Total deaths, yes. But again, the underreporting aspect is also very important. And so that's a big number, right? That's a really big number. But that number is very much in line with the projected number of people who are going to die because of drug-resistant pathogens by 2050. It's about 10 million people, and it will cross cancer. And there are many reasons for it. And I will now start talking a little bit about why it's projected so high, and what are the challenges when it comes to drug-resistant pathogens. So before I do that, it's a book by Malcolm Gladwell, Tipping Point. He calls tipping point as a phenomenon, or whatever it is, that once it crosses a particular threshold, it's difficult to stop it. Okay, almost becomes like a snowball effect. And I think when it comes to drug resistance, I think we are in that phase, unfortunately. And I mentioned we are in the post-antibiotic era. So what do I mean by that? The last class of antimicrobials were invented or generated almost 40 years ago. Okay, whatever new drugs you see are mostly side chain modifications of existing class, and there's a very simple reason for that, and it's economy, right? So if you are, the CEO of Pfizer, and I'm your R&D chief, and I come to you and say, hey, give me a couple of $1 billion, I want to, come up with a new class of antimicrobial, I have a brilliant idea. A couple of things, it's just going to maybe take 10 years for it to go through FDA. And it's very much possible the very next day the pathogens may become resistant, right? As a CEO of a company, you just don't see enough, return on investment, where there's always this challenge of, pathogens becoming resistant. Fluoroquinolones is a good example. Ciprofloxacin, for example, it has to bind to an enzyme called DNA gyrase. When the transcription process is happening in DNA, there's something called supercoiling, and if that enzyme with the ciprofloxacin will go and bind, prevents it from acting, then there's torsional stress and DNA destruction happens. But a single point mutation, just one nucleotide change, and the affinity of ciprofloxacin binding to that enzyme goes down 10,000 times, and all of a sudden becomes useless. And so that's a huge challenge. I think the other thing we all have to keep in mind is, I give the example of what's in a salad bowl, is the globalization aspect. So, you may consume a salad without paying attention where the ingredients are coming from. And sometimes I think it's very important to keep that in mind. You must have heard about the Wendy's, salad outbreak, tomato salmonella, for example, spinach outbreak. And so one thing we have to keep in mind is when you look at the US food supply, we are importing, food products from many, many different countries on a daily basis. The constant need that I want to eat that food 365 days a week, but somebody has to grow it somewhere, right? And so that is something to keep in mind. If you look at the latest statistics, in 2022, this year, more than half of the fresh fruit and almost a third of the fresh vegetables Americans buy is imported. That number is significantly going to go up by 2027, okay? And so how are we going to ensure that products that we are eating over here, with the assumption that they are raised in the US, have they met the standards wherever they are raised? So the Food Safety Modernization Act that came up under President Obama made sure that any product that's coming in the US is regulated by a third party, the same USDA standards that we have over here. So there are some good things that have happened from a regulatory point of view. But just to give you an example, always somebody is going to sneak through it, and then becomes a huge problem. Okay, so why is this important? Why am I stressing on the aspect that we bring in food products from different parts of the world? So from a pathogen perspective, here's a very good example. Salmonella ricin is a type of salmonella that was not there in the US. And we first discovered it in around 2008 because it was in white pepper that was imported from Thailand. In Thailand, this ricin has been, we know it's there, it exists in pretty high numbers. So it came to the US in 2008 in California and quickly started to make its way through the country. And then in 2010, it picked up resistance to 3 different antimicrobials. Okay, and when the original ricin was there in the US, it was not resistant to any antimicrobials. It picked up 3 resistance genes along its journey through the US. And then in 2013, we discovered it in pigs in North Carolina. Okay, so as you can see, it's just making its way through. And the multi-state outbreak of multi-drug resistant salmonella ricin infections linked to contact with pig ear dog treats, it's happened in 2019. Okay, so I'm just trying to sort of take you through this journey of how these pathogens come, they trickle through the country, they were never seen here, pick up resistance genes, and they start causing problems. So what happens when a pathogen that was not resistant picks up resistant genes? So there are studies looking at, when pathogens pick up resistant genes, the pathogenicity increases, the ability to, sort of get rid of the competition that they have, it improves their fitness, okay? So I'll tell you a little bit more about it. So here's another example of, this is a multi-drug resistant salmonella particular strain. When I say multi-drug resistant, I do not mean that it's resistant to 3 antimicrobials. I mean, it's resistant to 3 or more classes of antimicrobials. Okay, so this particular one was detected by the CDC, very low levels. So I can see about 2007, 2008, hardly we were able to find it. And something happened in 2008 and 9, it became resistant to 4 antimicrobials, right? The moment it picked up that resistant gene, it just shot up. And you can see after 2009, right now, it is actually number one cause of human salmonellosis in the US. Okay. Previously, as you can see, all the way from 1996, when the Food Net Services really started the surveillance system, it was not something to really worry about. So something happens when resistance comes in, improves the fitness of the strain, ability to cause infections, and so I'll talk a little bit more about it. So before I start talking about all this resistance and stuff, I want to just take you through what are the common mechanisms. And it's fascinating. There are many, many different mechanisms. I like this one that just came out a couple of weeks ago, in Nature, and a very nice pictorial as to what happens. So if I just take you through, the different class of antimicrobials, there are different modes of action. The idea is to kill the bug in the end, right? So some of them, if this is a bacterial cell, will come and just make holes in the cell wall. All right. So, I mean, just imagine if you make holes on your body, you're not going to survive. And, so pathogens are smart. They will make changes in the cell wall. Most of the mode of action is affinity. Like, this drug has to come and bind to something in the cell, in the pathogen to really start acting. Well, they'll make changes in the cell wall. If you can't bind to the cell wall, then you're useless. Or if they make it through the cell wall, so there are many drugs that have to get inside. If they get inside, there's something called efflux pumps right here. It's just like a pump. Something comes in, it'll just pump it, just go out. Okay, before it can really reach a level where it can kill a pathogen. And efflux pumps are very interesting. So efflux pumps are not always active, but there are certain things that make it active. Obviously, antimicrobials is one, but disinfectants and quaternary ammonium compounds are the other aspects that activate the efflux pump. So you may go and clean the surface and you will think, well, it's absolutely clean. But just think about it. If you have an E. coli on the surface that's got the efflux pump, you're not only activating the efflux pump, you're killing the E. coli that does not have the efflux pump. And at the end of the day, you're only having resistant E. coli on the table. Okay, so you're basically wiping out their competition, making it easier for them, and so on and so forth. They'll change the binding sites. So I gave the example of fluoroquinolones where they cannot bind to that DNA gyrase enzyme, and so it's a lot of it is also affinity based. So those are mechanisms of resistance. Now I'm going to talk about transmission of resistance. So an E. coli with a particular resistant gene can give it to another E. coli. Okay, so the important ones are over here are called vertical dissemination of resistance genes. So your parents gave you some genes, you will give some genes to your kids, and so on and so forth. That's vertical; it's going straight, and that's absolutely fine. It's the horizontal dissemination which is the big problem for us. So what happens in vertical is E. coli is giving to E. coli, right? In horizontal dissemination, E. coli is giving to Salmonella, okay? And that's a huge problem, right? And so pathogens are very smart. They will build this repertoire of genes. It's like, I got more missiles than you have. Simple as that, right? And so the more missiles you have, the more powerful you are. And vertical dissemination is through plasmids, for example. Plasmids are extra chromosomal DNA material in the cell that can replicate on their own, and plasmids are circular, so they carry a lot of genes, and they can make a copy and then through a process called conjugation give it to the other cell, and now both of them are resistant. So vertical dissemination is something that's really, really problem. One of the questions students typically ask me, so when did these genes start to evolve? When did we start seeing them? And Alexander Fleming, discovered penicillin in the 1920s, and is that, well, that's not the case. Genes have been around for billions of years, okay? Billions and billions of years. I can come to your backyard and take a soil sample, I'll find something in there. I've done that with my technicians, and they're like, wow, I got Salmonella floating in my backyard. You will find it. You will find it. And so you can see, some of these genes I'm talking about here, 1 billion years, 2 billion years. And this is based on carbon dating and, really going in areas that are not inhabited by humans for millions of years. So they've always been in existence. So now let's talk about, the connected world that we live in. I gave you the example of Salmonella ricin from white pepper, right? Now let's look at some really interesting cases. NDM1 stands for New Delhi metallobeta-lactamase. I think as researchers, one of the things we have to do is we have to stop calling these genes based on the places where we find them for the first time. This really did not make the Indian government very happy because you got a drug-resistant gene based on their capital is not good. But this was a new mechanism of resistance, new gene that epidemiologists and researchers, observed that, people in Europe have this new gene, and then people in India and Pakistan have this gene. So when they went to talk to these people, especially in Europe, they found out that everybody had a travel history to India within the last 1 year. So what was happening was they were going to India for cheap surgeries. Okay, this gene had emerged in the hospitals. So they picked it up from the hospital, brought it back to the country. Okay, and this can happen very quickly. So this is around 2008, 2009. And this was in Klebsiella, which causes pneumonia, and it's a big problem in the hospitals. 2012, there was a lady in Baltimore who had a UTI infection. They had an E. coli that had this gene, and she had no travel history. So in this 3-4 years of time, this gene had already jumped from a Klebsiella, which is mostly in hospitals, to an E. coli. And E. coli is like the DC metro station. Okay, everybody comes in, exchanges, goes, and we all have E. coli, right? And E. coli stock has also evolved. It's become smarter. It's an indicator pathogen, right? So we'll test some food product, and if we find E. coli, that basically means it's fecal contamination, right? Something in the processing stage has not worked out well. And so that means sanitation, cleanliness process. But now E. coli is also expanding its repertoire. And we have to also remember this one big family called Enterobacteriaceae that has E. coli, Salmonella, Shigella, Klebsiella. all the gram negatives that cause a lot of human infections are also causing challenges in the animal side, big reservoir in the environment. And anybody who's in the same family, they share a lot of, similar genomes. So exchange is also very easy. Okay. So let me give you an example of North Carolina. So this is a study we did in my lab. And this is, again, very, something that we didn't expect. Valerie Nelson was a DVM student, and we have a program in our college where DVM students will spend a summer to get some research experience. So typically, to learn new methodologies and technologies. So I was like, all right, here's a bunch of human isolates of Salmonella, go and play with it. So she characterized them, did some sequencing, a month and a half, presented a poster, she's happy. And I was like, that's great. And then a postdoc comes to me and is like, hey, Sid, did you see the sequences that Valerie had generated as part of the project? I'm like, I thought she was done. It's like, no. And that whole then discussion led to this publication with the, North Carolina State Public Health Lab. So what did we find in this one? Let me tell you the story. So you see the word mobile colistin resistance. Whenever something says mobile, it means it's on a plasmid. It moves very quickly. So I'm talking about the horizontal dissemination aspect over here. So colistin is a drug. It's been in existence for many, many decades, but typically not been used because it's nephrotoxic. Okay. But if your physician says either you die or you take colistin because nothing else is working, you're going to take colistin, right? Colistin resistance was always, the gene was always present. We knew about it. It was on a chromosome. So it's not mobile. When anything on a chromosome means it's vertically disseminated. Anything on a plasmid means it's going to go sideways. In 2016, a study came from China where they said that this colistin resistant gene has jumped from a chromosome into a plasmid. So we're going to call it mobile colistin resistance, right? So this was studied in 2016. It really caught everybody's attention because we are afraid this is almost our last line of defense. And now because this gene is jumped onto a plasmid, so it's going to start moving around from E. coli to other places. And physicians will already have a heartburn because of that. So coming back to the study that we were doing, these isolates were around from 2014 time period. Okay, these are human salmonella isolates that we get a copy of it from the State Public Health Lab as a part of a collaboration. So we found this mobile colistin resistance in this patient or in this sample. So obviously, the first thing we did was we went back to the State Public Health Lab and could you tell us a little bit more, which is always a challenge because of HIPAA rules and confidentiality. But they told us this was an 18-year-old female who had just traveled to China. She spent some time there. When she came back to North Carolina, she felt sick. She was admitted to a hospital. We isolated salmonella, and then we just put her into a freezer. And so we got her out of the freezer. The point is, even though it was first reported in 2016, this was already in the US in 2014 time period. Okay. And so that tells you that even if you find something later on, that doesn't mean that that's what's happening. This was already circulating in the US, and we don't know where all it went. But talking about, connectivity with different parts of the world. And that's what I mean by saying that when it comes to resistance, you can have all the amazing programs, surveillance program, I run 2 in my lab in the US, but it has to be a global approach because pathogens and, pandemic has made it very easier for me to explain my thought process that we are all connected together. So a little bit complicated. So when we isolate the plasmids, because they have to generate the evidence, and we, when we do the genomic sequences, we can start comparing it. And we found it was 100% matching with the same plasmid that was reported in that paper from China. And then we went back and then we saw that there was another strain in human in 2013 that was maybe around 95, 98% matching, who had a travel history to Thailand in 2013. So it was even in existence even before we thought it was. And who knows how long this has been floating around. Okay, so that was sort of big picture take-home messages, connected, pathogens can evolve anywhere, can come, so a little bit about mechanisms of resistance, vertical and horizontal dissemination. Now let's talk about, a solution. Why don't we, just take the antimicrobials off the market? and see what happens. Will the pathogens revert back to a stage when they were not resistant to any antimicrobials, right? So the thought process is the pressure on the pathogen by these antimicrobials is the Darwinian concept. Either they have to evolve or they die. So when they evolve, that means they're becoming resistant to their antimicrobial. And this whole thing moves very fast. So a pig can pick up a unique salmonella strain from the environment, from the floor, and start shedding that same strain that it picked up from the floor in 30 minutes. It's a very fast process. And we have to understand, in a test tube, when you grow E. coli, there are billions and billions of E. coli, and they replicate, become double the number in a matter of 17 to 18 minutes. It's a very fast process. They have a very straightforward genome. It's easier to make mistakes in that genome. And when they make mistakes, most of them are deleterious mistakes. They'll kill them. But some of them are good ones, and they like to keep it. And they are the ones that we should be very concerned about. So back to a susceptible population, is it that easy? So here's the thought process. You have a bunch of pigs, never been exposed to any antibiotics. When you look at what they are shedding in the environment, you will find some E. coli. There should not be any resistance to it. If you fall sick, you get treated, you're fine, you go back home. But let's say these pigs are now exposed to antibiotics. And because they're exposed, they're going to start shedding resistant pathogens. You fall sick, drugs are not working, and all of a sudden, your condition is. So this is the example I talked about, 3 to 5 days more in the hospital these people have to spend. So what if we stop giving antimicrobials? Do you think they will go back to this stage? And the answer is, I don't think so. Okay, so I will, because there's a big role of the environment in this. Okay, all the surveillance program that you see, even the ones that we are doing, they're very focused on the commodity. What's in that pork chop? What's in the chicken breast? What's in that human? Very few surveillance programs are focusing on what's in the environment that we all share together. Okay, there's no surveillance program that consistently globally looks at soil samples or wastewater samples, right? And I think that's the missing piece. So I put a dotted line over there because now I'm going to split sort of the rationale for the next few slides in these two different sections. Simply taking off antimicrobials, does that revert to a situation where pathogens are now not resistant? Okay, because there is no pressure on them. That's the first one. And the second one is the role of the environment. So let's look at the US data. So Campylobacter is a pathogen that you find in the poultry production, and it causes campylobacteriosis in humans. The number one risk factor for campylobacteriosis is consumption of poultry. Okay, so this is the latest US data. You can go to the CDC website, play with the numbers. It's a dynamic sort of a platform. But in around 2000, so fluoroquinolones, which is ciprofloxacin I talked about, were used in the poultry industry in the past to treat colibacillosis. Okay, so serofloxacin and enrofloxacin were used in the past. Then they noticed a study in Minnesota where a lot of humans who had consumed chicken had a Campylobacter in them that was resistant to ciprofloxacin. Okay, and then they figured it out that it's coming from the chicken industry because the Campylobacter in the chicken is exposed to these antimicrobials. They're becoming resistant, and because consumption of poultry is the number one risk factor for Campylobacter in humans. So an FDA judge in 2006 decided, okay, you told Bayer and Pfizer, you're not going to be able to sell your, Sera and enrofloxacin in the poultry industry, with the idea that if you don't expose Campylobacter in poultry to these drugs, they should revert back to a situation where they were susceptible to these drugs. But just look at the data. Actually, it's going up. It's not going down. Okay, and so the question is, well, you took the antimicrobials off. We should have seen that it go down. It's actually going up. So what's the reason for that? So now I'm going to start talking about the fitness of these strains that are resistant to antimicrobials. Okay. So here's a simple study that was done, and are drug-resistant pathogens better fit? Okay, and I will also explain a little bit more about what I mean by better fit as we move forward. But here's a very simple experimental study, a lab-based study on animals. So you take a bunch of chicken, you give them, expose them to Campylobacter that is not resistant to fluoroquinolones, right? Next day, you sample the fecal matter, you will find Campylobacter that is not resistant to fluoroquinolones. Next group of chicken, you give Campylobacter that's resistant to fluoroquinolones, and 24 hours later, lo and behold, you find resistant Campylobacter jejuni in the fecal sample. When you mix both the type of Campylobacter and give it to the chicken, the next day they found only the resistant pathogens that were shed. The susceptible population was gone. So there's a lot of competitive exclusion and fighting between the different types of Campylobacter for the same resources. And they found out that the jejuni that was resistant to ciprofloxacin was much more fit to compete for these nutrients compared to its counterparts, which are not resistant. And so this is very important. So what this tells you is that, and keep in mind, there is no antimicrobial pressure in this model. It's not like the feed had antimicrobials, nothing. It's absolutely clean. The only thing that was added were these different strains of Campylobacter, one with resistance, one without resistance. So I hope you will appreciate this slide because I put a lot of effort in this one. The famous Titanic one, right? So I think the antimicrobial resistance is just the tip of the iceberg, okay? Because the same genes that provide resistance against different antimicrobials, the pathogens are really smart. They are using the same genes for a lot of different activities, okay? Signaling, competitive exclusion, they're basically genes that they're using not just to prevent getting killed by the drug, but in all of other activities also. So basically they're using these genes to improve their overall fitness, right? It's like, you're not just active in the gym, you're also playing sports outside, and you're carrying big, heavy backpacks with you, and you have no problems at all in facing the world. So let's look at a current study that, how much time do I have for now?
It's 1:08.
So, okay, all right. Yes. Is organic production. So we go to high-end restaurant and you want that, naturally grown chicken, you pay more because, the concept is healthy grown, they're exposed to the environment. And since they're not given any antimicrobials, they should also not have any drug-resistant pathogens, right? So is that really the case? So this is a quick study we're doing comparing commercial poultry that's indoor to backyard poultry that's outdoor. But one thing to keep in mind is even major big commercial poultry industries are also not using any antimicrobials. So that's one thing to keep in mind. So why study backyard poultry? Well, the biggest outbreak that just ended in the US that involved 48 states was with backyard poultry, Salmonella. Okay, 2 people died, thousands of people sick, 1,000 plus people sick, many ended up in the hospitals. And one of the reasons is if you have to, hold a chicken in your hand, to hold a chicken in your hand in a commercial farm, you'll have to go through a lot of hoops and a lot of, okay, before you enter farm. Somebody's backyard, hey, you can just go in the backyard, pick up chicken, and that's the link over here. Is very close proximity to animals. So what did we find in these animals? So this is the resistance profile of E. coli, but it's extended spectrum beta lactamase E. coli. So what it's a mouthful, I know, but ESBL E. coli, this E. coli is different from the regular E. coli in terms of its expanded repertoire of resistance genes that it has. So your second generation cephalosporin, third generation cephalosporins, the ones that when you go, the physician is going to prescribe you, this E. coli is resistant to all of those. And you can see with the red bars, anything that's in red is resistant. So by default, they're resistant to first, second, third generation of cephalosporin. That's why the red bars are so long. But look in the backyard poultry. There's a lot of action in the backyard poultry. If I can move my cursor. Imipenem, meropenem, gentamicin, cefpenem, actually higher resistance than the commercial poultry. And so where are they getting it from? They're probably getting it from the environment. So you will wonder, what is ESBL E. coli doing in my backyard chicken, right? Environmental reservoir. Now, ESBL E. coli is, this 1980s in the US. It's a big concern, not just in the US, but also for the World Health Organization. Okay, their big surveillance programs is centered around ESB locali at the global level, and so it's really, really important. Okay, so now a little bit of complicated data, but I'm going to take you through it because you will be able to appreciate it more. This is looking at Campylobacter in North Carolina for 2 years, looking at the farm, looking at the processing stage, and then the retail stage. Okay, so that's what an animal typically goes through: raised on the farm, gets processed in a processing plant, and then ends up in the retail store. Okay, so this is a big study, and so I went back, sorry. Yes. So again, and of course, I'm not going to take you through all the approaches we use, but but before I start talking about, show you the key slide and show you what happens. So this is the entire state data. Okay. So now let me just a little bit complicated. As I talked in the beginning, I'm going to go at different levels. So this is slightly at a granular level, right? So a clonal complex, what's a clonal complex? It's basically sort of a family tree, let's just call it. So just like your family tree is going to be different from my family tree. You'll be more closely, ancestry.com, right? You got your own set, I got my own set. When it comes to pathogens, the thought process is that every particular pathogen has a founding genotype somewhere billions of years ago, I don't know. And we call it a ST1. What does ST1 stand for? It's a sequence type. It's a part of the genome that we have sequenced. And you take a sequence and then you merge it with check it with the entire database, and it'll say your sequence is matching with sequence type 891. So the advantage is if 891 was also reported in China, then I can say what I have in the US is what was in China. So that's the thought process. But the idea is that you start with the founding genotype, and just like your grandfather was different than your father, because they accumulate some changes and so on and so forth. And eventually what happens is they accumulate so much change, they start looking a little different from their founding genotype. So we call them ST2s, right? Then eventually over a period of time, this ST2 will start to further diversify. I'm talking about your granddaughter, and we call them ST3. So eventually over a period of time, they will also become ST4. So they're just accumulating genomic changes, and so they keep evolving, but they are still part of the same ancestry. They're still, and that ancestry is something we call the clonal complex. Okay, but eventually something happens that this ST4 will diverge so much that you can't call it ST5. You're going to have to call it ST50 because it just something happens. It just picks up so much change. And remember, I gave the example of us, but we are talking about pathogen in this case. It's a faster, and that becomes clonal complex number 2. Okay, so now the reason I took all this time to explain this is these are the different sequence types we detected at those 3 different stages. Okay, so one is and the 2 species of Campylobacter, jejuni and coli. Jejuni is causing majority of infections in humans. Remember, chicken consumption is the #1 risk factor. Coli causes very few infections in humans. But this slide explains to you the competition that happens at different stages. So I always ask everybody in the audience, can you tell me 2 cool things about this slide? And I'm not talking I'm just talking basic stuff.
No coli in these stores.
Bingo, that's one. What's the second one? There's a lot more action in the processing stage. You see how the numbers significantly go up? And the reason for that is very simple. Processing plant are the one where chicken from different farms are coming together. So they bring their own population, and there is just huge influx of these strains. But what is interesting is coli was there till at the farm, at the processing, and then at retail is just gone, right? And that is also replicated in the infections caused in humans. 90% to 95% are caused by jejuni through poultry consumption because coli just vanishes. So it could be because of the processes used during that phase to that coli is not able to survive. Could be some other factors also, right? And so, but it's interesting. Now, a little more complicated, but Promise, this is my last complicated slide. I talked about the, clonal complex 1, clonal complex 2. So you can see all the jejunites are in that corner, or the coli's are on the other side. What's the interesting thing happening over here? Do you see that bridge in the middle? That's the coli and the jejuni merging with each other. Okay, that's the one I was talking about. That sometimes they change so much that they move far away, but here the jejuni and the coli are coming together. And this is important because on an average, coli are more resistant to different antibiotics. Jejuni has a much better affinity to call infection in humans. So imagine you create a Campylobacter that's not only resistant, but it's also causing more infection than humans. And all of a sudden you have a winner, which is not good for us. So what's the solution? Should we not eat food animals and start eating, lab-based meat products? And I just got this on CNN. FDA has given a safety clearance to a lab-grown meat from cultured chicken cells for the first time. And this particular company is just waiting approval from USDA. And then once it gets approved, they can start producing stuff and you can buy it. This chicken patty costed, the total cost of the chicken patty was around 175,000 pounds. Okay, they had to grow these in the cell cultures. To give it the color, they used beet juice. And I think he had to chew this at least 26 times before he could swallow it. But now we are in this phase where cell technology is improving. A sequencer that used to cost $300,000, you can get one for $100,000. A thermocycler, 30, 40 years ago nobody could afford. I remember I made a special trip to a lab to see a thermocycler for the first time. I remember that. So now I'm coming to almost towards the end of my presentation. And this is the analogy I give to my students when it comes to resistance and the fitness and everything. So when you go on a hiking trip, there are some, challenges when you go hiking, right? You got to carry a big hiking backpack with you. It's got a tent, it's got food, water, tent or, things that you can, comfortably stay because the environment is harsh. The analogy is the harsh environment is the antimicrobial. And this big backpack, heavy backpack is your resistance genes. Because resistant genes are heavy, they are difficult. They take a lot of energy for those pathogens to maintain them, right? So the idea is if you remove the harsh environment, which is my second image, that when you go back to school the next day, you are not going to carry that big backpack with you. You don't need it. Everything is, controlled temperature, why would you need a big backpack, right? So the idea is when you take those harsh conditions, pathogens will lose that backpack. But have you heard about the ultra marathon runners that like to run 50 miles, 100 miles? That's what these pathogens have become. And they love carrying those backpacks because they've improved their fitness so much. They're like, it's almost like I'm showing off. I can carry a big backpack and I can run 50 miles. So that's what's happening over here. So they're carrying multiple backpacks, which is multiple different genes. I just created this slide last night. I think you get the idea what I'm talking about over here. So it's improved fitness, and it's amazing. Okay, so I want to round off my presentation by this particular slide where I'm going to sort of recapitulate everything. I call it the AMR conundrum. It's 3C. I call it: challenge, the complexity of the problem, and how we can work collaboratively to solve the problem. So it's a global issue. I think we talked about it. I showed you many examples of pathogens floating around the world. Accessibility to drugs. I showed you that picture where the gentleman was buying, drugs as if he's buying, vegetables in a supermarket. And that's unfortunately is the reality. I'll give you a very good example. I was in Malawi a couple of months ago. One of the fellow travelers got an eye infection. We went to a pharmacy store and she's like, you can get it from there. That corner is all the drugs we have for eye. Went there, got an antibiotic, walked out. And guess what? He was fine the next day. Economics, I talked about the example of, approaching a CEO, it just doesn't work out. And that's a challenge. Globalization, I talked about, your salad bowl. Hey, you want to eat this, 24/7. Well, somebody has to grow it, right? Population, we just touched 8 billion people. In a couple of decades, we'll be 10 billion people. We have to increase our current agricultural practices by 70% to feed that world. Okay, 5 grams of protein consumed in 1960, 25 grams now. Where are we going to get that? We can't do it backyard, right? So intensive production. Well, how do you do intensive production? Complexity. I talked about the reservoirs. These genes are everywhere. I did not talk about ecological niche. So it's interesting. Salmonella has 2,500 different serotypes. But whenever there's an egg-related outbreak, they always look for Salmonella enteritidis, okay? Because enteritidis has made the egg as its home, and it will not let any other Salmonella survive. And only you will find enteritidis in eggs. You can go and do a Google. Egg-related outbreak, Salmonella enteritidis, right? Ecological niche. It's like, it's my house. You're not supposed to come in here. Fitness of the strain. I think I talked a lot about, how it improves the fitness. Evolution, survival of the fittest. Organic system. I think it's a complex. I think it's a good system. There are some advantages, but still a lot of work needs to be done. Backyard production, for example, is not regulated by USDA. Okay. So just like your backyard is different than my backyard, similarly, people are using all kinds of things. And so education is very important. Okay. One Health, I think this is a classic One Health problem. I think ownership is very important. I think we have to take ownership, whether it's the human side. I've been guilty of, my physician saying, you got to take this, pill that's going to leave a really bitter taste in the mouth 3 times a day and for a week. And if by day 4, I'm feeling fine, I'm like, God, I don't want to, eat anymore. But what you have done is you have basically killed the ones that were susceptible. The resistance one that are still sticking over there, you basically remove their competition. And that's why you have a relapse. And the relapses are even more difficult to treat than the first time you have it. Judicious use is very important of antimicrobials. And that has to be a global effort. I know WHO is working very hard to make sure antimicrobials are used judiciously, but we still have a long ways to go before we reach that stage. Stewardship is important, and so is awareness. I cannot stress how important awareness is. Education, all the way from food animal to human side to environmental side, and for people from different parts of the world. The education aspect is important because it's such a complex topic, and so many different varieties in here that it's difficult to get hold of one thing and say, I got this, I can solve the whole problem. It's not going to happen. If you can figure it out, let me know. Okay, thank you so much. All the work has been done by people in my lab. I just do the talking, but I have an amazing team of students and technicians, postdocs who do all the work. So kudos to them, and also I have to thank all the different funding agencies. Thanks again for patient listening. Appreciate it.
I have one online question, which is that now we are told to raise the temperature of various foods to specific temperatures to render them safe. Do pathogens evolve resistance to their vulnerability to high temperatures? Yes, so there have been studies doing on it, in terms of impact of, say, water content, for example, where pathogens can survive. You will always find exceptions. And so that's why you will also see, USDA coming up with different recommendations, that they will change the recommendations to, make sure the temperature of inside of a pork chop is 140 degrees. I don't know exact what it is, but I would not be surprised. Studies have done where there are strains that can resist really exceedingly higher amount of temperatures. Our key thing is to make sure that they don't spread and don't spread their thermotolerant properties to other particular bacteria. But I think one of the things that I've learned is we have to respect these pathogens, because we don't see them. Maybe sometimes we don't appreciate what we don't see. We really have to respect them. And the pandemic is a very good example. 5, 6, 8 million people have died. They have been on this planet for way longer than we have been. So they know the system. You talk about, where most of the drugs are coming from. Many of them are coming from soil fungi, streptomycin. So, they produce them, but they don't get killed by it because they have defense mechanisms, and they talk to each other, they exchange stuff. So it's a huge challenge. Yes.
Yeah, as you talk about the economics, it was I think in the late 20th century, the entire vaccine market was something like $3 billion.
Mm.
And now Moderna has a market cap of $100 billion.
Yeah.
So obviously an extreme example. But do you see, and given the rapidity of the process that the mRNA, messenger RNA
Mhm.
approaches exhibited, do you have any optimism for having more personalized approaches so that you can take a sample from someone and then more or less real time come
Yeah.
up with?
So personalized medicine, point of care medicine is big and getting bigger. Okay, so for sure. But I think you will have to look at the expense of all of these things, you know. I think, and I think, so we have vaccines against, you know, you can get a vaccine for malaria. You can get vaccine for traveler's diarrhea. But the challenge with the pathogens is we are not dealing with one type of, you know, SARS-CoV-2. Salmonella alone has more than 2,500 different varieties or serotypes. So in the food animal production setup, you know, we have vaccines for different, like E. coli, you know, F18 strain or K88 strain, depending on, you know, stage or what age of pigs you're using. But it's tough. It's tough because of the diversity of bacterial pathogens that you see. Okay, and you know, you may react in a very different way than I, you know, to a particular pathogen. That's the other thing. You know, they don't have one particular site that they go, like, you know, COVID, you know, yeah, you know, your lungs are busted, right? These guys can go anywhere. And the problem is they typically stay in the gut. So if you have E. coli food poisoning or something, you know, your physician is not supposed to give you any medication. Only thing you're supposed to take is lots of water, lots of fluids, and take it easy. But in case of resistant infections, that becomes a problem. Okay, and if you look at the history, you know, these pathogens are gaining more and more genes. I did not talk about, you know, stuff like you know, integrons that can carry up to 200 to 300 gene cassettes on them. Can you imagine being infected by an E. coli that has an integron with 200 drug-resistant genes on it? It's a problem. It's a huge problem. So I think the diversity of pathogens that we see makes vaccine are good, but it's just tough. It's just tough, and it's expensive. It's expensive.
Any other questions? We are just about out of time.
Okay, great.
Yeah.
Thank you.
Okay, thank you so much. Sid. We
All
appreciate
right, thanks.